Hi, this is Miss Linton, and this is 7th period review. Say hi.
Hi.
For Honors Bio, Unit 5, Stranger Danger. Okay, let's talk about the different big ticket items that you want to make sure you know how to do it, okay? So let's start with Unit 5. The first chapter is Chapter 5, okay? So in Chapter 5, donde está? There it goes. All right, the big ticket items that you want to know is you want to know the cell cycle. Could you draw a cell cycle for me? Do you know how to do that? Well, what do I do? Here's my circle. Now what? Okay, but G1, S, G2, what are those all part of? Interface. Okay, so I would have this big piece of pie, and all of this would be interface. And then interface I could break into what?
G1.
G1?
G1's S and G2.
Yes. G1, S, and G2. Okay, then this stage right here is the M stage. But the M stage consists of two things. What two things?
Mitosis and meiosis.
No. Mitosis and
Okay.
cytokinesis. And mitosis consists of what?
Prophase,
Yes.
metaphase,
Prophase,
anaphase, and telophase.
Yes. Okay? Now, knowing what happens, have we gone over that? Could you review what happens in each part of that? Yes, you can. And you know what everything looks like. All right? Then the next thing I want to know about is how, not on this cell cycle, not only what is it, but how is it controlled? How can you control a cell cycle? What could you use? What could you do? Yes?
Like the modifiers or the moderators?
No, I'm looking for something else. What controls how many times your cell reproduces or doesn't reproduce? Signals, right?
Okay.
And we talked about that? Like a growth factor? Exactly. So you have signals that can help control it, that can turn the cell cycle on, and I can also turn the cell cycle off. What do we call it when that cell is done? In fact, it's so done I want to kill it off. What is that called? Apoptosis. Apoptosis. We have two stranger dangers. What classes are you in? You're just interested? Go. Nice. Emma, good friends. All right. Then the next part. Any questions on that you want to go over? Oh, could you give me examples of apoptosis?
The web fingers.
Yeah, the fingers, right? Death here. Okay, good. Death here. Okay, next thing we need to talk about in Chapter 5, we talked about chromosome structure. And did you not have a additional objective on that? How the DNA winds on those histone proteins. Remember that? Wasn't there an additional objective on that? I'm pretty sure there was an additional objective on that. Yeah. Okay, so you're going to want to review that. You're going to want to review numbers like haploid and what? Diploid. Okay, how many chromosomes do we have? 46. So our diploid number, 2n, equals 46. That's our diploid number. Diploid means two. I have two sets of chromosomes. Half my set came from my mother and half came from my father. Okay, that's the diploid part. Half mom, half dad. A total of 46. But when making sperm or egg, I only want what? 23. Because I want to take 23 from a sperm plus 23 in an egg, join those together in fertilization, right? And make a diploid what? Zygote. Cleavage, 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 right? Okay, so this process by which you go from 2n down to n is called what? Meiosis. Yes. Meiosis, right? But if I'm just going to go from 2n to more 2n, what would that be called? Mitosis. Mitosis. Now I'm going to tell you right now, there are plenty of cells, we don't talk about it yet in this unit, where they go end to end. Haploid structures making more haploid structures. If it's end to end, what would that be? Mitosis or meiosis? Mitosis. When it's same, it's mitosis. If you bring it down by half, that's when you're going to have meiosis. Yes?
Meiosis just completely separate from the cell cycle.
It will happen in a cell cycle in your either ovaries, for you ovaries, or testes. We have it in specialized areas where our cell cycles will, this will not be mitosis, this will be meiosis.
So goes through interphase and then meiosis, just in different
Meiosis 1 and meiosis 2, yes. All right? We good on that? Next thing you need to know. You need to know stages of mitosis. If I said to you, draw me a cell in the stages of mitosis with 5 chromosomes, could you do that? With 5, could you do it with 4 chromosomes? Could you do it with 2 chromosomes? How would you do that drawing? What are the stages of mitosis? Mitosis. What are they? Prophase.
Metaphase, anaphase, and telophase.
Good. So prophase, what's happening during prophase?
They're just chilling.
Nuclear envelope is
Yep.
Nuclear envelope is starting to disappear. What else? Okay, so hold on. Whole thing's called mitosis. First stage, prophase. The chromosomes become visible. The nuclear envelope and the nucleolus disappear, radiating microtubules. Esters. They meet in the middle at metaphase, away at anaphase, to nuclei at telophase, cytokinesis. Did you not model that with chromosomes for me?
Licorice.
Yes, you did. Yeah, you're licorice. Okay, so you should be familiar with this. So let's do, what did we say we're going to do? 5 chromosomes?
Yeah.
Okay, so prophase, the chromosomes are starting to become what? Visible. And they consist of what? Sister chromatids. So I'll have 1, 2, 3, 4, 5. Are they doing synapsis? Are they doing any snuggling? No, snuggling's for what? Meiosis, getting ready for what? Sex. Okay? Mitosis, my arms, my legs, mitosis, not getting ready for sex. You only snuggle when you're getting ready for sex. Right? Okay? So now we could also, I know, that was great. Okay? Here's some little centrioles over here. We could start to have, you know, our spindle fibers starting to form. There's our prophase. I would label it prophase. I would point out that that is a chromosome. I wonder why I'm spending so much time doing this. I would be going things like that. These two are sister chromatids, right? Those are the kinds of things I would be doing. Okay, what stage follows prophase? What is it?
No, metaphase.
Okay, so here's metaphase. What happens during a metaphase?
They meet in the middle
At metaphase. Are they meeting up with their homologous pair?
No.
No, that's with what? Meiosis. Okay. So 1, 2, 3, 4, 5. Can I see all 5 chromosomes? Can I see that they're lined up? Okay, do I have a nuclear envelope anymore? No. Could I have spindle fibers getting ready? Yes, I could. What stage follows next? Away at anaphase. So I'm going to pull them across, away from each other. 1, 2, 3. Pulling them away on anaphase. Yes? And then what would telophase look like? Yeah, and I would still be able to see them. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. And I'd be starting to form my nuclear envelope again. But I could still see all 5. Would I draw them at this stage like this again? Would they miraculously have a sister chromatid again? No, why not? The sister chromatids have been what? Separated from each other. Okay, good? All right. Next thing you want to know about, prokaryotes do it differently, right? Prokaryotes cannot do replication like that. Prokaryotes have to use what? Binary fission, single circular chromosomes. You want to review that. You're going to want to review your stages of meiosis. There is meiosis, what? 1, and there's meiosis 2. Okay? So, what key event occurs during prophase 1? That's when homologous pairs are snuggling, exchanging sister chromatids. Good. What key event occurs during metaphase 1?
The homologous pairs meet.
Homo middle, which you now know from our last chapter, what?
Independent of.
Yes. Beautiful. Wonderful. What happens in anaphase 1? Who?
Pairs break apart.
Who breaks apart?
The pair.
Homologous pairs. Okay? Homologous pairs separate. Okay? How many cells do I now have? 2. Okay? Any key event in prophase 2? No. Is there any more crossing over going on?
No.
No, why not?
They're not.
There's nobody to cross over with. The homologous pairs have been separated. You'd be crossing over with a sister. No real variety there, right? Any key event happening? What happens in metaphase 1?
2.
I mean, yes, that is what I mean. Metaphase 2. Thank you, sweetheart.
That's when the sister.
Yeah, then so the single chromosomes are in the middle, right? So here, they're single file, right? Like this. Whereas here, they're like this, yeah? Okay, what happens in anaphase 2?
The sister chromatids separate too.
Yes, sister chromatids separate. Good. What happens in telophase 2? 4 cells. Right? You're gonna have 4 cells. They, from here down, they're considered haploid. Why? Because they don't have their homologous pair. So they're haploid at this point. Okay, and then you could compare mitosis with meiosis. For the last thing on chapter 5 is you want to know the difference between spermatogenesis and oogenesis. Do you remember what the big difference was there? Spermatogenesis, ooh?
One makes sperm, one makes egg.
Yes, one makes sperm, one makes egg, but there's more to it.
Versus uneven separation.
Uneven separation in females, remember? Males, if they're going to do a meiotic event, they're going to make 4 sperm. Females, we're going to make 1 egg and 3 little polar bodies. 1 egg and 3 little polar bodies. Okay, chapter 5, done. Next.
Miss Lynn,
Yes.
does anything happen in prophase 2?
No. I mean, chromosomes are visible, but there's no crossing over anything other than what we would expect. Okay. Now, for your chapter, what is this, 22, right? Chapter 22, you want to know, it's all about the stages, okay? So I want to know what happens following fertilization. What is the next stage after fertilization? Tell me.
Zygote.
Okay, all right, put that in there. Zygote, good. Tell me something else. Cleavage, cleavage, which is a bunch of mitosis, right? You just keep doing mitosis, right? Okay, wait, don't jump ahead. Wait for me to ask, okay? And as a result of that, you will form a what? No.
A
A marula, which is a what? Solid ball of cells, okay? Then what comes next?
Blastula.
A blastula, which is a what? Yes. How do you get a blastula though? Pump and salt. Who follows? Water. Blastula. What do you call the cavity inside the blastula? Blastocele. C-O-E-L. Okay, what comes next? Gastrula. You're letting out the gastrula. Okay? And what is a gastrula? Layered ball of cells. How many layers do you have? 3. What are those 3? Meso and endo. Do you know what becomes of those 3 layers? What's ectoderm? Skin, nervous system, lens. What becomes of endo? Resp and digestive. And this is everything else, whatever's left out. whatever's left over, right? What do you call inside the endoderm when you're looking at that tube, then there's mesoderm, ectoderm, what do you call, this is going to become your gut. But it starts off, that inner container is start off, you call it the, yep. And remember, depending on that cavity, what the opening becomes, it's either going to be the mouth or it's going to be the anus. For us, the opening for us is the anus. The blastopore becomes the anus for us. For others, the blastopore becomes the mouth. Okay, what stage follows gastrula?
Nerula.
Nerula. And this is the beginning of your spinal cord. Okay, and how does the nerula form? Because the notochord induces the epidermis above it to fold in. Remember that? So that is a great example of induction. So words you want to know are things like induction, morphogenesis, and morphogens, differentiation. Remember with differentiation, we said you have one zygote, this cell. This zygote is going to become trillions of cells, trillions of cells. How do you get trillions of varying function cells from one zygote? Because they're all going to have the same DNA. Do you remember? What was it? What was different? What caused differentiation?
Oh, the proteins in the
Proteins in the cytoplasm, affecting what genes? So how do you get differences in proteins? Well, we said where the sperm penetrates, that's going to be the anterior end. Remember this? And the other, by default, is going to be the posterior. And then in cells that have more yolk, what's that yolk region going to be?
The opposite of dorsal.
You're right. Okay, so this is dorsal.
Vegetal.
Vegetal pole. And vegetal pole, and this is the ventral front. Do you remember what pole this is called? Animal. Animal. Okay, so right away, as soon as we start having cleavage, you have differences. Because this end right here has just sperm. This has sperm and yolk. This has yolk. Nothing. So we have differences right away when we start doing cytokinesis. And it's what's the morphogens, it's the proteins surrounding the DNA of these trillions of cells that can influence what traits are expressed. What do we call those genes that control other morphogens? Do you remember?
Homeotic genes.
Yes, homeotic. Homeotic. Okay? Those homeotic genes will code for other morphogens, which will trigger other homeotic genes, which will trigger other morphogens and start you down your differentiation pathway. Okay, then the last part of this chapter is, as far as humans go, it's what happens in week 1, week 2, week 3, week 4, week 5, week 10, months 3 through 4, months 5 through 7. Could you answer those questions? Tell me week 1. Blastula, and you begin implantation. Tell me week 2. Gastrula. Week 3. Nerula, and what else? Heart. 4. Limbus. Senses.
Wait, isn't that? What about gender?
Find out.
Oh, we're coming up. What's this one?
Placenta.
Placenta. What's this? Gender. And cartilage starts to become bone. And what's here? Survive.
Hey, you're out of the
Okay? Now, let's talk about something else. Birth. Stages 1, stage 2, stage 3. Something comes out every time. What's stage 1?
Water break.
Yeah, amniotic sac
Doesn't the cervix also?
and cervix dilates. It doesn't break, but it dilates. Okay, how about here? Who comes out?
Baby.
Baby. Who comes out?
Placenta.
Placenta. Good. Aging. We had theories on aging. You remember what those were?
Genetic.
Genetic. I live long, my parents live long, right?
Stress factors was the third one.
Hormones
Yes.
start slowing down.
Hormones, whole body is hormones. Whole body is all about the hormones. And then what?
Environmental factors.
Yeah, extrinsic factors. Take care of yourself. Got it?
Yes.
I can't believe I wrote that fast.
That was really good.
Thank you. I work hard. Okay. Next thing we need to know, chapter 23. Okay? You need to know things. Oh, let's see what I want to start out. You want to know chapter 23. Got to know about Mendel. I believe you have an additional objective on him, right? So know about him, a little bit about his history, YPs.
Because they're easy
I know,
to-
just do it. Okay? He had 3 laws. What are the 3 laws?
Segregation,
Dominance.
Okay.
dominance, and independent assortment.
Okay. Do you need me to explain those?
Yeah.
Okay, so law of dominance says, okay, if I have this individual right here, law of dominance would say that they will be tall and they will be round. Why? Because big T is dominant over little t and big R is dominant over little r. Okay? Law of segregation would say when I'm going to make gametes with this individual, I could get big T with big R, or big T with little r, or little t with big R, or little t little r. Law of segregation is saying, oh crap, sorry, back up. Don't write that. I was getting ahead of myself. Oh, stop, don't. Okay. Law of segregation says when I'm going to make my gametes that I'm either going to give a big T or I'm going to give a little t, not both. Right? I'm going to give big R or little r, not both. That's law of segregation. Law of independent assortment says when I make my gametes, I could get combinations. Big T, big R, big T, little r, little t, big r.
So gametes can have multiple letters, but genotype can't.
No, our genotype has multiple letters, but the law of dominance says in an individual, tall is dominant over short, round is dominant, let's say, over wrinkle, okay? But when I make segregation, when I make gametes, I'm either going to give, because of anaphase 1 and anaphase 2, I'm either going to give big T or I'm going to give little t. I'm not going to make any gametes that have both, because then I'd be making diploid gametes, not haploid gametes. Yes?
Can you explain like the different types of crosses, like dihybrid and like?
Yes. So monohybrid cross would be like this, big T with little t. That's a hybrid, this is a monohybrid cross. Mono, 1. Dihybrid, that would be when I have something like this going on. Right? So that's when I have, 16 squares going on here. Because I'm doing, let's say, big T, big R, big T, little t, big R, little t, little r, right? Dihybrid, I die 2, right? I would have to make, for a trihybrid, I would need 64 squares. We don't ever need to do any of that because we do the product law. And the product law is, say, take your individual punnets and multiply them together. Yes?
Can you do a practice problem?
Sure.
Yes, thank you.
Do you have one you want me to do in particular? Do you want me to do a trihybrid cross? Is that what you're asking for? And I want to know, what are the odds of making a baby that's like this?
In the book, instead of giving us the parent, it like just gives us traits for the parents. Can you explain how to do
Sure.
that?
If you have a book, pick one, and I'm happy to do it. Okay, so how would you do this one? What are the every single time this one's going to give a little a, right? And half the time this one's going to give a big A, and half the time it's going to give a little a, right? So the chances of little a would be what? 1/2.
Yeah.
Okay, in this case, every single time it's going to give a little b. Half the time this is going to give a big B. Half the time this is going to give a little b. So again, little b would be 1/2. This case, every single time they're given a little c, half the time big C, half the time little c, so 1/2. 1 times 1 times 1 is 1, 2 times 2 is 4, times 2 is 8, 1/8. Okay? All right, now, period, what period?
4.
4. Cool.
Awesome. Thank you.
You got it, babe. Okay. All right. So let's listen to this. So in order to do 2, I actually have to do 1 first. Okay, because you use stuff from 1 in order to do 2. So, question 1 says, attached earlobes are recessive. Okay, so you have attached and unattached earlobes. So when I get questions like this, I write them out. So big E, anything, and there's little e and little e. I'm picking E for ears. And so it says attached earlobes are recessive. So little e, I'm going to say that is attached. And big E, anything, they would be unattached. Okay, whenever you read, write it into some sort of genotype. Okay, it says, what genotype do children have if one parent is homozygous recessive for earlobes? Homozygous recessive for earlobes. How would I write that?
Little e.
Little e, homozygous recessive for earlobes, and homozygous dominant for hairline. Hairline, what they're referring to is big W anything is widow's peak. So little w would be what? Straight. Okay, so he said homozygous, let's see, one parent is homozygous recessive for earlobes and homozygous dominant for hairline. So what would homozygous dominant be for hairline?
Big W.
Right. You all right so far? Okay, and then it says, and the other is homozygous dominant for unattached earlobes. Homozygous dominant, homo meaning same, right? So what would that be?
Big E.
Big E. And then they said, and homozygous recessive for hairline, what would that be?
Little W.
Little w. Okay, every single time, what is this person going to get?
Little W.
Big E, little W.
Big E, little w. And every single time, what's this person going to get?
Little E, big W.
So when this happens through fertilization, what are you going to have?
You're gonna have unattached earlobe.
Big E,
Big E.
little e,
Big W, little W.
big W, little w. Do you see that? Here, let me put a number on here. This is practice problem 3-1. Got it? Okay, now I'm going to get rid of this. All right. Now, the number 3, oh, yeah, that was 3-1. Then 3-2, it says, if an individual from this cross reproduces with another of the same genotype, so that would be, we just figured out what it was, right? We just did it. What are the chances that they will have a child with a straight hairline and attached earlobes? Straight hairline is what?
Little W,
Little bit. And they said attached earlobes? So that would be what? Little e. So, here's the parents. Here's mystery. What are the odds? So then I just need to do individual punnets. Can you see this punnet? Yes? Okay? And then I would do the W's, and the W one is going to look just like this, but with W's, right? So what are the odds of little e, little e? What are the odds of little w, little w? 1 times 1 is 1, 4 times 4 is 16. You good on that?
Wait, hold on.
Okay. 4 o'clock. And I have to pack up. But if I do it by 3:15, I should be okay. I haven't picked out what I'm going to do yet. So I'm interest. 100% sure. Okay. We good? Got it? All right. Okay. Now. Okay. So let me keep going on here. That would be an example of a dihybrid cross. Okay. Then let's review this last part. Oh, we did product law. Oh, let's talk pedigree. Okay, that's the next part. So pedigrees, and if you recall, I suggested to you. Here, I'll drag it over. Maybe. Come here. All right, perfect. Okay, so now let's talk about this. Okay, I want to know which of these traits would work with autosomal dominant, autosomal recessive, or sex-linked. Could you do that?
Can we practice it?
Yes, we can practice that. Okay, and the way I suggest you doing it is just working your way through all of them. So, autosomal dominant. Remember the colored in one and sex-linked. The colored in ones have the disease, right? Okay, so if it was a dominant disease, we have to have at least a big A here and a big A here, and they don't have what? Except your little a, little a. You all right with that? Okay, so now this person right here, they could be giving them this big A, and if all they have to give is a little a, would that work? Do I even care what the other one is?
Nope.
No. So would this first one work with an autosomal dominant? Yes. Now I'm not memorizing this because memorizing it, then I can't apply it to a new pedigree. You don't know what pedigree you're going to get. I'm just showing you the logic behind it. Okay? But this one works with a dominant, so I'm going to put a D next to it, all right? Let's try this one. Will this one work with a dominant disease? No, because if they have it here, whoops, sorry, let me do an A. If they have it here, one of the parents would have to have it, and these guys, because they don't have it, right, it would not work. So as far as dominance goes, that's a no-go for this one. You see what I'm doing? Okay, what about this one? Would it work for autosomal dominant?
No.
No, why not?
Their kids are normal.
Do we skip generations with autosomal dominant?
No.
You can't.
You can't skip generations, right? Again, take a look. If you ignored this and just looked at this, that looks a lot like that, right? It wouldn't work. Because you can't have a disease popping up here that one of the parents didn't have if it's a dominant disease, right? So on this one, dominance is a no-go. Okay, now let's do autosomal recessive diseases. So new disease, okay? So in this case, if I had a disease, this could be little a, they have the disease, little a, they don't have the disease, what could they be?
A.
Big A, little a, will that work?
Yeah.
Yeah.
Why?
Because they've both got an A.
Right? So this will work with a recessive disease. Okay? Will this one work with a recessive disease? What is it when you have the disease?
Little A.
Okay, is there any way they could each do that?
Yeah.
How?
Little A.
Big A,
Each
little A.
one.
Exactly. So will this work with recessive?
Yeah.
Yeah.
Yes. Okay. Will this one work?
Not with dominant.
We said not with dominant, but will it work with recessive?
Yes.
Yeah, this could
Yeah.
be little a, this could be big A, we don't know. This one could have a little a, we don't know, and a big A. They just don't have the disease. You could make a little a, and this one could have one of their big A's. So will that work with recessive? Yes or no?
Yeah.
Yes. Go ahead.
On the quiz, will you tell us whether we're solving for dominant or
No, that's why I'm problem solving with you right now on how do you tell if it would work for a dominant or recessive.
So how would you, like, what would the question be?
I would show you a pedigree and I would say pick which one of these it will it work with.
Oh,
Just like I'm doing now.
So we figure out whether it's dominant or recessive or both or not.
There's one more. Sex-linked.
Excellent, yeah.
Okay, so let's see if this would work with sex-linked. Now, if a woman has this disease, right, a woman would have to be X little a, right?
Yep.
Okay, her mom would be X little a. Mom could give one of these. Where's this one coming from?
Daddy.
Does dad have the disease? No. Dad doesn't have the disease, so that must mean he has a big A. So would this work with sex-linked?
no.
No. So sex-linked on this one is a no. Okay? Would this one work with sex-linked?
No.
Is that a male? No.
This is a male, so what would he have to be?
X A Y A, right?
No letters on the Y. X, little a, and Y. Dad doesn't have the disease, so what would he be? And Y, right? This is a boy. What is he getting from dad? Just the Y. So this would have to come from mom. Could she have a genotype that would provide a little a?
Yeah.
What would it be?
X
big A, X little a. And so this little a would come here. Remember, all the sex-linked diseases that I'm giving you are what? Recessive. Recessive. So this would work right here with X-linked. Would this one work with X-linked? Dad would have to be what?
X little A. X.
Mom is at least a X big A. We don't know about her other one. They have a girl. Does she have the disease?
No.
No, so she has to be a X
Big A.
big A. That would have to come from mom.
Yeah.
Because dad, the only X he has to give her is a what?
X.
Little a.
Yeah.
Okay, she marries a man. He doesn't have the disease. What is he?
X big A Y.
X big A Y. To his son, he would give the Y. Could the mom give this little a she had from her dad? Yes. To the daughter, he only has his good X to give. Oops, sorry. He only has the good X to give. That's all he has to give right here. The mom can give whichever one she wants. We don't care. She doesn't have the disease. So would this work for X-linked? Yes. Got it?
Yep.
Is this
Okay.
on YouTube?
Yeah, I'm recording it right now. Okay, the only other thing we didn't talk about, you need to know the difference between multiple alleles, okay? Multiple alleles, an example of that, the good example from multiple alleles is blood. Things like big IA, big IB, little I. Those are multiple alleles. Okay? Codominance, that you see in blood type. Both alleles expressed. Incomplete dominance. That would be like H1, H1 is straight, H2 is curly, and then your example of incomplete dominance would be an H1, H2, which is what? Wavy.
Oh, like the pink and the red.
Yeah, red and white makes pink.
How do you know whether one's
Go ahead.
Dominance versus, are you just gonna tell?
Codominance would be you have some straight hairs and you have super curly hairs.
Okay.
But wavy is like a mixture. It looks like blending to the eyes.
Okay.
Incomplete dominance looks like blending to the eyes. Okay? X-linked inheritance we already just went through, right? And then polygenic would mean that your trait, whatever you have, is equal to, you know, it could be equal to R's and Q's and P's and L's, anything along the, giving you that one trait. And that was things like skin color and height. Those are polygenic. Intelligence. Remember we said intelligence isn't big S, smart, little s, stupid, right? It's multiple genes. Yes?
How come for the incomplete dominance, it's like the 1 and the 2's on the bottom, and for the dominance?
They're just written that way. Sometimes these numbers will actually be up here, so don't let that throw you off. I know you're looking for a pattern, and I would too, but sometimes they actually put those up there.
All right, thanks.
Okay, and then the last one is, we talked about environment. Environment are especially like to work on polygenic, and that was the rabbit with the ice pack, right? And his hair grows in black. Or I told you the sex of like alligators and stuff can be determined by the nest temperature. And then the last thing was linked genes. And what that means is you won't have independent assortment with linked genes because those genes are on the same chromosome. So they will travel together. No independent assortment. Yes? Unless crossing over occurs.
So, can you go over polygenic once more?
Polygenic is, it's not like height of a pea plant was big T you're tall, little t you're short, right? Our height is not determined by one pair of alleles. There's several alleles that contribute to our overall height or overall intelligence.
Okay.
Okay? Do your practice problems, do your additional objectives. All right, that's it. Good job, you guys. Oh my gosh, 42 minutes of fun.